All right, chapter 16. So this is the last one. Yay, we're almost done. So chapter 16 is on the sense organs, so this will make this chapter a little bit more fun, dealing with a lot of your different senses in your head as well as the rest of your body. So sensation is a subjective awareness of the stimulus. So having those receptors receive that stimulus information and then sending that stimulus information to the brain where we can actually be consciously aware of that information. So sometimes your sensory information is filtered out in the brainstem. So for example, a type of gating situation that prevents information we don't need. So stuff that we can habituate to, stuff we don't need to pay attention to. Sometimes even pain could get filtered out in the brainstem, and therefore we're more likely to continue on and less likely to feel what's going on. Some signals actually don't require conscious awareness. So for example, pH and body temperature, our body just fixes that without us having to be like, okay, we need to cool down, so I want my sweat glands to do their job. Your body just takes care of it for you. So your sensory receptors transmit 4 different types of information. So your type of a stimulus, where that stimulus is coming from, how strong the stimulation is, so intensity, and how long the stimulation is lasting, so duration. So for modality, the type is usually produced, like for example, your vision, hearing, taste, touch. That's your type or modality of your sensory information. For location, telling you where that sensory information is coming in. So depending on the receptive field of your neuron, you could either have very fine detail of where that information is coming from, or just get a general location of where that's coming from. So for example, on the back of the person, the receptor field is very wide for a sensory neuron for touch. And so if I put this little kind of protractor looking deal on the back, it can give me a general information on where that information is coming from. But if I move that same protractor pointy thing to the hand region where the neurons have a smaller receptive field, then you're able to tell the difference where each of those little pinpricks are coming from. So here, all the information is getting sent onto one neuron, so kind of give a general information, hey, they're both coming from this area. Here, you're able to locate more specifically where those little pinpricks are coming to. So smaller receptor field, therefore having a finer detail on that sensory information. So with your location, that information from those individual neurons are then sent to the brain and mapped onto a specific location. So their projection pathways go to their particular mapped out destination in the central nervous system that tells your brain, hey, this sensory information is coming from this particular location. Next, for intensity, so how strong the stimulus is, it's based on which fibers are sending the signals, how many fibers are doing so. So more fibers sending that sensory information, telling the brain, hey, this is a bigger bigger stimulus than, for example, another one. And then how fast they're firing. So for example, if I were to lightly touch my hand, that's going to have a soft type of slow cadence for signaling to my brain. However, if I pinch my skin, then that's going to send a faster signal to my brain that the brain determines, hey, that's pain, that's a more intense stimulation, and something needs to be done so we're not in pain anymore. The last one is duration, so how long it lasts. So for some senses, you can actually adapt to that sensory information. So the signal that is sent is still sent, but it gets slower over time. So you have 2 different type of receptors and how they adjust for sensory adaptation. So you have your phasic receptors that adapt fairly quickly. So for example, smell, movement of the hair on your skin, as well as cutaneous pressure. So the seat that you probably are sitting on now, you, for the most part, have been able to ignore the fact that, you're still getting that touch sensation on your butt for the chair and your butt connecting. However, just because I just told you, you're now paying attention to that sensory information now. Same thing with smell. You know, you go somewhere, smells not that great, or your little air freshener, you're like, oh, that's a nice new smell, and so therefore, you know, eventually over time, you stop paying attention to the nice smell. For your tonic receptors, they adapt more slowly. So this more so focuses on your proprioceptors, so dealing with body position, muscle tension, and joints. So just maintaining kind of a little slight awareness to that sensory information so that as your body is moving, you still have that kind of, oh yeah, in the back of your head, yeah, that's still there, in order to be able to make the appropriate response for maintaining your balance or doing different types of movement. The receptors also have their classifications. So we have by modality, so the type of stimulus, so receptors that detect heat, so thermoreceptors, photoreceptors detecting light, nociceptors detecting pain, chemoreceptors detecting chemical signals, as well as mechanoreceptors detecting movement. Also based on where the stimuli is coming from, so exteroreceptors for outside or external stimuli, interoreceptors for internal stimuli, and our proprioceptors for body position and movement. And then lastly, by distribution. So our general senses, which are widely distributed through the body, or our special senses, which are limited to the head. So what we normally think of our five senses, like vision and hearing and taste and all that jazz, that's limited to the head. General touch senses is touch and pain is spread out around the rest of your body. So first general sense is pain. So pain is a discomfort that's caused either by tissue injury or the threat of tissue injury, so leading to what's called evasive action, so removing yourself from that painful situation. So even if you know your muscles are sore, and so you try to stretch out your muscles in order to relieve the pain, same thing, just trying to avoid that painful situation. Or if you're, slouching in a chair and you realize, oh wait, I do need to sit up. And therefore, your back is going to feel a whole lot better. So the receptors that receive pain signals are your nociceptors, and there's two types of them. You have your fast pain signals. So this is a sharp, localized stabbing pain, usually with injury. So quickly telling the brain, hey, something's wrong, please, we need to fix this. Or your slower pain signals, or slower nociceptors, for more of a diffuse feeling type of pain. So something that's a little bit more, kind of more of a soreness than a very kind of like sharp pinching type of pain. There are two locations where you'll receive pain signals. So the ones that we pay attention to are our somatic pain, so from our skin, muscles, and joints. The other type is visceral pain. So this is located in your viscera, so your internal organs. And these receptors are either for stretch or some kind of chemical irritants or ischemia of the visceral. This type of pain is poorly localized because for the most part we don't pay attention to the sensations that come from our organs. So we're not paying attention to how our food is digesting in our stomach. That's something we don't need to pay attention to. So when that pain signals are sent to the brain, it actually gets a piggyback ride onto another neuron, and then causes the brain to got to be confused on where that pain is coming from. So for example, when you have a stomach ache and you just kind of grab a random section of your stomach based on where that pain's coming from, it's due to that visceral pain kind of getting a piggyback ride onto another neuron that's going towards the brain. So these receptors receive the pain signal based on chemicals that are released from injured fibers. So these tell those nociceptors that, hey, something is wrong. In order for us to detect pain from regions where we normally aren't paying attention to the sensory information coming to that area, this is called referred pain. So in our little picture, the guy's having a heart attack, so the pain signal is coming from the heart, but because, again, it's hitching a piggyback ride onto the brain, both of this area, so his kind of chest region and some parts of the left arm are in pain. this one's like, okay, I don't know where this is coming from, but hey, let's get to the hospital, because any kind of chest pain, you want to hurry up and get to the hospital. So because it's signaling on the same neuron that you're normally receiving the information you're normally paying attention to, so your somatic pain or somatic sensation coming to that same neuron, the brain is kind of confused on where in the world this is coming from. This is the convergence of neural pathways. So brain doesn't know where this information is coming from, so it just automatically thinks, oh, well, I'm used to touch on the outside of the body, so it assumes that that visceral pain is coming from the skin. So this is why he's clutching his arm or his shoulder when the problem is actually his heart. So here's a mapping of different locations for referred pain. So again, heart in the left side of the chest, as well as part of your left arm, lung and diaphragm in this region, liver and gallbladder from this region. Again, is based on the convergence of those neural pathways, and they end up projecting on particular areas. So some of them, they're close to where they should be located. This one technically not, but oh well. Liver and gallbladder, so we do have it in the area where it should be. Stomach is kind of in near your stomach area. Appendix, near your appendix. Colon, near your colon. So still close to the regions where they're located for that pain, but still not the exact location of where the problem is. In order to deal with pain, we can give the body, or the body also produces, what's called an analgesic. So these analgesics are chemicals that will relieve pain. There's painkillers called endogenous opioids that the body makes itself. So endo, like inside. So inside, made by the body, painkillers. And those two types are your enkephalins, as well as your endorphins and dynorphins. Basically, again, their job is just to kill pain. These are secreted by your central nervous system, the pituitary gland, so that gland that just can't hang at the bottom of the base of your brain, your digestive tract, as well as other organs. So making their own painkillers so that you're not getting that pain to the brain. They can also act as neuromodulators to block pain and give pleasure. Another way to moderate the pain coming from your body is through spinal gating. So in your spinal cord, the fibers that are in the brainstem block the pain signals from what's coming from the spinal cord. So descending through the reticulospinal tract, so it's just one of those pathways in the spinal cord. So going down this pathways, helping to block the pain signals that are coming up to the spinal cord. So basically just protecting you from actually feeling the pain that is normal. Another way to activate spinal gating, so instead of these analgesic fibers stopping the pain signal, you can also rub or massage the injury. So again, working out, you will tend to massage that area to help, make yourself feel better, but really you're activating your spinal gating in order to get rid of that pain signal. Another thing happens, like for example, when you're hit your finger with a hammer and you start shaking your hand. Basically, similar concept to activate spinal gating, but also in order to confuse the brain on where that sensory information is coming from. So basically, still stopping that pain signal from consciously telling your brain, hey, something's wrong. So those rubbing helps to stimulate the interneurons to secrete the enkephalins, which are, again, another analgesic or painkiller. Moving on to the chemical signals, one being taste, the other being smell. So focusing on taste for right now, the stimulant for it is the chemicals that are inside your food. So as you're chewing up your food, the food gets broken down to smaller and smaller bits. They flow over your taste buds. Some of those taste buds are part of your the bumps on your tongue, so your lingual papillae, to help sense. Taste buds for taste, lingual papillae for food texture. So here in the back, helping to determine the actual food texture of what you're doing. Also having a lot of taste buds concentrated in this back area, so right before you swallow. So teeth break down your food, and those smaller bits are able to get into those taste buds, so you're actually able to taste your food. So inside your taste buds, you have taste cells that are actually going to receive the taste signals. And then, for example, also, of course, they're exposed to the outside environment, so you have your basal cells in those taste buds to also replace your taste cells when necessary. So on our tongue, we have our high concentration of texture and taste buds at the back. You still have taste buds all along your tongue. So zooming in on one of those taste buds. So your digested food gets dissolved in the saliva. So this is why you want to take your time chewing your food so that you actually can taste it. So that saliva, about to get disgusting here, is going to seep into the taste buds where you actually will move to zoom in for another higher degree of magnification. Sliding into that taste bud, filling into this taste bud hole where those sensory fibers will send that information to the brain. So in order to be tasted, your food molecules must be broken down enough to dissolve in saliva and then flood into that taste bud. There are 5 primary taste sensation, and they do have regional sensitivities on where you're tasting stuff. So moving your food around to the different areas in the mouth, making sure that you actually can taste what you're eating, and so therefore actually able to enjoy what you're eating better than just, okay, slamming on one side of the mouth, chew, swallow as fast as I can. So here's your 5 taste sensations. You have salty, sweet, umami, which is a meat taste, sour, and bitter. The spicy taste that is in a lot of foods is actually not an actual taste. It's due to the capsaicin or other chemicals in that spicy food that stimulates pain receptors, not taste buds. So what you're actually getting is just a little bit of pain associated with other flavors for whatever you're eating. So still adding to the taste sensation that you're getting, but still actually it's a little bit of pain. This is why when you get something that's super ridiculously spicy, it's just painful and no longer enjoyable. For your taste pathway, it moves from the cranial nerves into your brain stem, where it sits to your hypothalamus and amygdala. So these are 2 brain stem structures for helping to control your autonomic reflexes for either salivation, as in, okay, I'm tasting something I like, I want to go ahead and break it down even further so that I can taste more of it, as well as make sure that I break down whatever I'm going to eat. Also helps with gagging and vomiting. So part of that is also for people who have like texture issues, or for example, they just don't like the taste of certain stuff, and then it automatically causes them to gag or vomit what they are eating. It's because it's sent to those 2 locations, and those locations are like, nope, we don't like this, get rid of it. So therefore gagging to either get it out of your throat or vomit to get it out of your stomach. The other location is the thalamus, which is superior to your hypothalamus. It sends the information to the cerebrum for our actual conscious sense of taste. So knowing what exactly that we're tasting and putting into our mouths. Our next chemical sense is smell. So the sense of smell is also called olfaction. So the response of the stimulus due to odorants or chemicals into whatever we breathe. So inside our nasal cavity, we have our olfactory mucosa, so covering up those concha that are on those bones that are inside our nasal cavity. So inside our olfactory mucosa, we have these fascicles that extend through those holes in the cribriform plate. So we covered that earlier on the sides of that kind of fin structure of the... Oh, I'm drawing a blank. Okay, basically that fin structure that you had to do for the bone exam. Anyway, so holes in the cribriform plate on the ethmoid bone. Those fascicles extend into the olfactory mucosa. They're actually exposed to the outside environment. So as we breathe in, whatever we breathe in, the little tiny particles, they activate these fascicles. Then the information is sent up to the olfactory bulb and then through the olfactory tract to the brain. We have about 10 to 20 million olfactory cells, again, in our olfactory mucosa. But we're actually able to distinguish between 2,000 and 4,000 different odors. So the coding that happens in the olfactory bulb help us to determine the actual smell that we sensed. Okay, so this is kind of more complicated, but still saying all the little bits and pieces. So whatever little particles that are in the air that we're smelling, those will stimulate those nerve endings. Those nerve endings move up through those holes in the cribriform plate and then into our olfactory bulb, do a bunch of synapsing, sending that information along. So our neurons involved with detecting smell, again, are the only neurons in the body directly exposed to the external environment. So because that, they have a short lifespan, only about 60 days, about 2 months. Because, again, you're exposed to the outside environment, so you're more likely to get damaged. So therefore, they're going to die off fairly quickly. So those basal cells that are nearby will differentiate into new olfactory cells in order to replace them. So when they die, they're still replaced, so you're still able to have a sense of smell. So for your signaling, for your odorants, so these are your chemical ligands binding to your nerve endings that are part of those fascicles off of your olfactory bulb. The odorants activate a G protein couplet receptor, and then that G protein moves off, eventually making our cyclic AMP, which is our second messenger. That eventually is going to trigger the action potential in order to send that information to the brain. The olfactory receptors adapt very quickly, so this is why I can go into a room or go into grandma's house and it smells like mothballs. It might take a while, but eventually you're going to adapt to that smell. So there is some synaptic inhibition in the olfactory bulbs, as in, okay, we smell this isn't not something new, so I don't have to pay attention to that. Some odorants also act on our nociceptors, so our receptors for pain, and those odorants being ammonia, menthol, chlorine, and capsaicin of peppers. So still same thing. Some of them kind of pleasant based on their level, so like menthol, kind of minty flavor, that's kind of cool. Kind of like that smell, or something spicy, that's kind of like associated with, hey, okay, that's something that's going to taste good. However, it's actually still just a slightly bit of pain that you're actually sensing. Ammonia, chlorine, just part of like different chemical cleaners. Okay. 362? Yep. Okay. So, olfactory projection pathway. So, from your olfactory cells, they'll synapse in that olfactory bulb. And then when it synapses onto a glomerulus in that bulb. So, it helps to code what the particular odor is. Then, that odor is sent to the olfactory cortex in the temporal lobe. Then, also sent to other locations in your brain. So, it's not just, okay, yeah, we smelled this. It's also associating smell with certain things. So, for example, to the hippocampus for memory, amygdala for emotions, hypothalamus, your insular cortex, as well as your orbitofrontal cortex. So, helping to tie the scent that you smell with whatever you're tasting, as well as with memory. So, hippocampus. So, if you're smell something like, that smells really familiar, or you smell something like, oh, that reminds me of grandma. Same thing. Tying it with memory, scent's actually your strongest one that's tied to memory. Again, emotions with scent, as well as different visceral reactions. Moving on to hearing and equilibrium. So, focusing on hearing, it's a response to vibrating air molecules. So, the vibrating air molecules are what's creating your sound waves that eventually hit into the back of your ear, and that energy is then transferred to the cochlea in your inner ear. For equilibrium, it's based on the movement of hair cells that are in the semicircular ducts still in your inner ear area. So both of these senses have mazes filled with fluid, and then the little sensory hairs in them help to determine that central information. So that motion for those cells is converted into a pattern of action potentials to tell your brain that sensory information. So first thing for sound, it's the high or low sound or the pitch of it. So something that is high pitch is very squeaky in sound. Something that is low pitch is more kind of like a bass drum type of sound, or like a lion roaring. This again is determined by the frequency of the sound waves or the vibration of them. So a bunch of frequency or bunch of sound waves per second is a hertz. The hertz is your unit of measure for your sound waves. So how many waves per second is in that sound? So human hearing range is 20 to 20,000 hertz. Speech is found at 1,500 to 5,000. Most hearing loss occurs in 250 to a little over 2,000 hertz. So thinking of female pitch in the voice, so higher pitch, this is why sometimes when, couples are older, the husband starts losing the ability to hear his wife's voice. It's because the pitch of her voice is part of his hearing loss range. Okay, for the other part of sound, it's loudness. So how strong of a sensation is that? So it's measured in decibels, and the sound waves are going to be taller in their size than a softer sound. So that helps to code on the loudness of that sound wave. With sound waves, of course, you have to be careful. So excessive sound over 90 decibels can cause damage. So down here at the bottom, we have a decimal scale. So softest sound that a human can hear is 0. Kind of our softer sounds like leaves moving, about 30 decibels. Libraries, 40. Normal conversation, 50 to 65. Moving up to louder stuff like your vacuum cleaner or your lawnmower. This is where you have 90 decibels for your lawnmower. So a lot of people will wear hearing protection while they're doing the lawn. Here is your moment of where you have instant hearing damage just from the exposure. Sometimes your headphones, if they're up too loud, they can cause you to have instant hearing damage in your ears. Jet engines, 140 decibels, so this is why your baggage people and people who are on the runway are always wearing hearing protection, just because a jet engine taking off is loud enough to cause instant hearing damage. So here's our location for the start of hearing damage. Anything above that, you're going to have increasing levels of damage. So internal structures for your ear. We have outside the ear, you have the auricle of the ear that helps to funnel the sound towards the ear, moving into our ear canal. So through that external acoustic meatus here, then to the internal one on the outer side, so that tunnel where it moves through. Hitting our tympanic membrane or eardrum, that eardrum will vibrate depending on the sound that is being heard. That energy is then transferred through your inner ear bones or your ossicles to the oval window on your vestibule. That sound wave energy is then moved to your cochlear, so this kind of snail shell looking section in the back of there. Other parts of your internal anatomy for the ear, you have up here, you have your vestibulocochlear ducts. So they're oriented in three different angles in space in order to detect head movement. Then lastly, all this information is sent on the vestibulocochlear nerve, which is a combined nerve from the cochlea and your vestibule region in this part of the structure. So sending that information through cranial nerve number 8 to your brain. Your body has its own way of protecting your ears from hearing damage. Of course, it doesn't completely do anything, but what it does is it helps to prevent hearing loss, at least slightly. So the ossicles, our inner ear bones, as well as the muscles that are attached to them, can lessen the transfer of energy to the inner ear, so helping to prevent damage in the cochlear and cause the inability to hear in certain frequencies. So this is called the tympanic reflex. The tensor tympani muscle pulls the so the other one is your tympanic reflex. The tensor tympani muscle pulls the tympanic membrane, so your eardrum inward, therefore making it more tense and therefore less likely to vibrate in in the situation where you're having loud noise. So lessen the transfer of energy, lessen the ability of responding to that energy from the sound waves hitting that eardrum. Other reflexes, part of your tympanic reflex, reduce the motion of your stapes, which is one of your inner ear bones, so less transfer of energy. And it helps to prevent the vibration from transferring to that oval window in part of your, towards your cochlear. So just prevents that information from getting into your brain.